How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're listening to DNA Today, a podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research, we have you covered. For a decade, DNA Today has brought you the voices of leaders in genetics. I'm Kira Deneen. I'm a certified genetic counselor and your host. Joining me now is world-renowned geneticist, Dr. Madhuri Hegde. She is the Senior Vice President and Chief Scientific Officer of Global Lab Services at PerkinElmer. Thank you so much, Dr. Hegde, for coming on the show and joining me on this episode.
Thank you for having me, Kira.
So why don't we start by just explaining some of the basics and explaining the difference between whole exome sequencing and whole genome sequencing before I have all of my questions about whole genome sequencing.
Yeah, I think that's a great way to start. So when we are talking of the genome, we are talking of all the genes that we know today, around 22,000 genes. The sequences within the genes which are not making the proteins, and the sequences between the genes as well. So we are talking of intragenic, intergenic sequences and the coding regions of the genes. So essentially that makes up the genome. When we are talking of the exome, we are really taking out that tiny piece of the genome about the coding regions of the genome, which is about 1 to 2% of the entire genome.
So it's such a small slice of that pie when you think about 1 to 2%. That's so tiny. And we used to say, oh, the rest of it is junk DNA, which is a term we don't use anymore because we know if there are changes in the rest of the genome, it certainly can have effect on our genes and how our body reads our genes and processes them. When is it important in the clinic to order a whole genome sequence? Does it ever make sense to just jump right to a whole genome sequence and bypass exome and genome panels, or gene panels, I mean?
I think that's a great question. I think to answer that question, we really have to see what has happened in the last 10 years. When I started in genetics, we were doing Sanger sequencing and we were doing single genes, right? Most of the testing in clinics is ordered based on the clinical presentation. That's one thing. The second thing is, at least within the US, there's a lot of dependency on whether a test is going to get paid or not. So that's one side of it. The other side of it is technology. The technology has gone so fast and so quickly that today we can do the whole genome in a very affordable price or cost rather in a clinical lab. So your question is very valid, but the answer is little two-pronged here, in the sense that it depends on the laboratory setting, the affordability of the test itself, and can it be done in that particular setting. And I'm talking global labs here because I run global labs and I can understand the constraints different labs face. Now, if you just look at the technology, I would say go straight to whole genome sequencing. Ideally, largely because you're sequencing the entire genome, you're getting all the coding regions, the intragenic and the intergenic sequences. Your diagnostic yield is going to be higher compared to the exome. But having said that, I would still say that exome sequencing is probably the right way to start based on the cost, the reimbursement, and the laboratory setups that are currently out there. There are not that many labs which are able to do clinical whole genome sequencing.
So we'll get there, but for now, I think, as you're saying, most of the time you're starting with exome, then depending on the clinical presentation, insurance coverage, where you are in the world and all of that. But you see that gene panels are not being ordered as much anymore because whole exome is replacing gene panels in some settings.
That is true. So it's a very interesting situation. In the clinical setting, in a clinic, in a genetics clinics or a pediatrics clinic, where there's an unexplained condition suspected and could be genetic, they are going to exome and genome. And gene panels are not really that highly ordered. But if you just now flip that, where we got so many therapeutics coming up, and large pharma, small pharma are getting interested in genetics and specific genes, they are not running exomes and genomes. They want to do a panel because they have specific genes they want to look for, and they will contain it to doing small gene panels based on what they are looking for.
And when ordering a whole exome sequencing, and providers are looking at lots of different labs that provide this, is there a difference between labs, or is it just a very standard test that it doesn't matter where you get it from?
So that's a great question, right? And it's a very important one too. In most of my talks, I always say this, that genetic counselors who are really in the middle between the patient and the lab, actually deciding the test order and where to go, I think they are very important questions. The physician and the genetic counselor need to ask the laboratory. First thing is the method used. How are the genes covered? Are genes, there's deep coverage of the genes.
So how many times a gene is actually read? You don't want it just once, you want it many times.
Correct. And you will see there is a difference. Some labs do 70x, a standard is 100x. Then what is the design of the exome itself is absolutely critical. And the reason I say that, I'm going to give you a very simple example. Okay, Duchenne muscular dystrophy, majority of the events are deletions and duplications, about 70%, and 30% are single nucleotide variations. Out of that 30%, about 5 to 6% are deep intronic changes. They are deep into the introns. Duchenne introns are very big, 50 kb introns. So unless your design is done in such a way that you are tagging that particular single nucleotide, then you're not going to pick it up otherwise, because you have not put a bait there. So depth and how the design was done is absolutely critical to look at when ordering exome sequencing or any gene panel.
Because you want to avoid a patient having, this patient's negative for DMD, and now we've missed this diagnosis, and Correct. The whole diagnostic odyssey is going to really be affected from that. So you don't want to misdiagnose.
Absolutely, and it's very difficult to order the same test again, especially when you're depending on insurance to pay for it.
They'll say, no, you already did that. I'm not paying for that again. And then they don't understand all of these details. As a lot of people in healthcare don't even understand these details that are ordering this. As you said, this is what healthcare providers, genetic counselors need to be asking to know, okay, well, which lab am I going with? What is it actually going to tell me? And what can the test not tell me? Both sides of it.
And I think the genetic counselors play a very big role in this. Nowadays, the training of the genetic counselors includes all this information. They are very much up to date with all the conferences they are attending, and they play a big role. They understand this well, and they are ordering the test in interest of the patient. And this is very important for that reason.
Yeah, as a genetic counselor, it's up to me of what lab I'm choosing, for my patients. And so, the patients aren't deciding that. Very rarely am I in that situation where I say to patients, we could do this or that. But another thing that I've always wondered with whole genome sequencing is, does it ever include mitochondrial DNA? Because that's not technically part of the genome from the nucleus. It's kind of its own genome in a sense. Is that always included? Sometimes? Never?
So when you're doing exome sequencing, you have to make sure you put the baits for the genome, for the mitochondrial genome, just to be correct. When you're doing whole genome sequencing, you're sequencing the autosome and the mitochondrial genome as well. So you don't have to do anything special for that in the whole genome. The beauty of doing whole genome over whole exome, especially if you want to look at the mitochondrial genome, is the depth of sequencing you get when you do the whole genome and the mitochondrial sequence. You can get 1000x depth, and the data is absolutely amazing to look at.
So you said before depth, some labs might do 70, but you said 100 is a standard, that should be 100, but you're saying it could be up to 1,000.
Correct.
Wow. That is
for the mitochondrial genome.
For the mitochondrial genome. Okay, that makes sense, because for the mitochondria, there's only so many genes there. It's not as large as the nucleus.
It's very small. It's 16 kb. It's very tiny.
Yeah. Very, very, just a fleck compared to the genome and the nucleus. Yeah. Have you heard of the Blueprint Genetics My Retina Tracker program? This program offers eligible patients in the U.S. no-cost comprehensive genetic testing and counseling for their inherited retinal degeneration. The My Retina Tracker program panel includes 351 genes, mitochondrial DNA, and non-coding variant assessment. Stay tuned for our interview with Blueprint Genetics about the My Retina Tracker program and the patient registry on a future episode of DNA Today. In the meantime, you can learn more at blueprintgenetics.com. Again, that's blueprintgenetics.com. Curious to take a peek inside your body's drug response? Then you should check out Picture Genetics' PGX test. Powered by clinical laboratory Fulgent Genetics, Picture's PGX test is easy to order and understand with hassle-free clinician involvement and good-looking reports. Plus, you're fully supported through live chats, emails, and genetic counseling. To order your Picture PGX test kit, use code DNA Today for 25% off and free shipping. Get actionable genetic insights today to benefit your health of tomorrow. Do you know the power of polygenic risk score? Polygenic risk score, or PRS, enables healthcare providers to more effectively help patients lower their risk of life-threatening diseases with the latest breakthrough in clinical genomics. Allelica has launched the first PRS test on the market to calculate ancestry-specific PRS for breast cancer, prostate cancer, coronary artery disease, type 2 diabetes, and Alzheimer's disease, and delivers the results in a clinical-grade report. The PRS test is a laboratory-developed test that is performed in a CLIA-certified, CAP-accredited genetics lab. Here's just one example of why this test is so important. If a patient with breast cancer tests negative for BRCA1, BRCA2 genetic mutations, Allelica's PRS test may be able to identify other key genetic mutations which played a role in the cancer development. Learn more about PRS in our previous interview with Allelica CEO and co-founder Giordano Botta on episode 168 of DNA Today. You can order the PRS test at order.allelica.com. For 25% off and free shipping, use code DNA Today at checkout. Allelica, empowering the next generation of clinical genomics. So when we're looking at ordering whole genome sequencing, how often does it happen where whole genome sequencing reveals an additional condition or disorder that the ordering provider wasn't expecting? That's not the reason they ordered the test. So like an incidental finding. So they ordered it because, if we're going with our DMD example, so we're looking at that, but we find out, actually there's, a hereditary cancer syndrome we're identifying. How often does that come up? Because you're looking at the entire genome, and that's a lot of information.
Exactly. So in our hands, and we started doing whole genome sequencing in 2017. So we were one of the first labs to offer whole genome sequencing on a clinical basis.
I think you were the first lab, right?
Yes.
Yeah. I was like, don't give yourself a little more credit than that. Yeah.
Thank you. Yes, that's correct. And we have seen, I have categorized it into several categories. One thing is, I'm just going to go through it, right? Dual diagnosis. About 5 to 10% of the times we have found more than one gene involved. There's another category I called the blended diagnosis, where we see two different conditions looking as one condition, but we find different reasons for it. Then there is another category which we have recently started using this terminology, is a camouflage, where it looks like something, but it is something completely different. And we find this quite often nowadays, because you're right, we are looking at the whole genome, we are tagging deep intronic mutations, intergenic changes, we can look at copy number changes, we can look at repeat disorders, SMA, there's a whole lot of information you're gathering from it. So we are in the process of putting our data together to publish it.
Wow, that's exciting, because I imagine you have so much data in terms of, when people order, like those 3 types of incidental findings that you could have from it. And really changing a patient's care, I imagine, because as you said, it's actually 2 disorders, but we thought it was 1, or there's an extra disorder we didn't even have on the radar, all of these different situations where I'm sure many of those cases, patient care is changed because of it.
Yes, absolutely. And one of the things which I have sort of seen the difference from doing whole genome, because we are getting samples from all over the world, just given the global footprint of our labs. We get a lot of samples from the Middle East, a lot of samples from India and Malaysia. And what we are doing is we are building up that variant data bank, if you can say that. And that really empowers us to interpret better. People from all over the world live in the US, and many times we find variants which are not represented in the Caucasian population. So having that database at hand really helps us get closer to the diagnosis.
Yeah, and that's so important. I've brought that up multiple times on this show that when we have genetic testing, that oftentimes if a variant comes up, if someone is of European background, they're much more likely to be able to figure out, okay, what does this variant mean? Is it just human diversity? It doesn't affect anything health-wise, or, oh, this is actually causing the condition that we're trying to look at. So it's fantastic that PerkinElmer is really playing a role in ramping up that database so that we don't have the disparity as much for people of non-European descent, because this is such a big problem. When we started in doing genetic data and looking at this, we started with people of European descent, and we're not including people of all different backgrounds, and there's just a major problem there.
You're absolutely right. I mean, I've got so many examples now, especially from newborn screening, where in the US, we have picked up some variants in Indian population or the Chinese population. And we have been able to talk back to our labs in India and China, where that variant is probably a common variant for a particular disorder. And one particular case comes to my mind is PKU, which is one of the most commonly.
That's how newborn screening started. Yeah.
Exactly.
Yeah, what, 1960s, somewhere in there, like decades ago.
Correct.
And you guys have been doing newborn screening since 1994. You've had 7.5 million newborns screened since then. So that's like 26, 27 years, something around there. So you're really a global leader in this newborn screening, as you said, looking at people from all over the world. How do you see newborn screening changing in the future? I mean, as we said, we've been doing it since the 60s in different ways. It's really changed over the years, but how do you see that changing in the future? Do you think whole genome is going to get incorporated at some point, or it probably depends on cost. That's probably the biggest factor. But
the way I look at it is, I think there's going to be an intermediate step, and that intermediate step is probably going to be a gene panel. And the reason I say that is you can control the cost significantly. Today, if you look at some of the techniques and methodologies that are available for doing hybridization and bait selection of genes, the pricing has come down quite significantly. So there is right now a discussion going on about newborn screening modernization. And I think the question is going to be whether it's going to be molecular first or biochem first. Obviously, molecular first will heavily depend on our ability to interpret data, the variants of unknown significance. We don't want to send families on a trip of something that is not even real.
You have to build up that database before we do it, as you've been talking about. Yeah.
So I think it's going to change. When it is going to change is time and cost is going to decide it. But gene panel of the RUSP genes, let's just take for that as an example, is definitely on the horizon.
Yeah, and hopefully that will find even more babies that have these conditions so that we can do treatment immediately. And that really changes the outlook of a baby's life. That's the reason we do newborn screening.
Yeah, I mean, Pompe is a great example, right? Neonatal Pompe and adult onset Pompe, we know the whole spectrum of Pompe. What if we can pick it up so early that the symptomatic event never really happens?
Yeah, especially for those very well-known disorders, like you said, like Pompe, we understand this condition,
yes,
as opposed to some other conditions where we say, well, we still need a lot more research to understand that spectrum, as a lot of genetic conditions are at that point. Polygenic risk scores, or PRS, are a powerful way to identify an individual's risk of common disease by analyzing up to millions of genetic variants spread across the genome. Allelica has launched the first PRS test on the market to calculate ancestry-specific PRS for breast cancer, prostate cancer, coronary artery disease, type 2 diabetes, and Alzheimer's disease, and delivers the results in a clinical-grade report. Order your test today at order.allelica.com. For 25% off and free shipping, use code DNA today at checkout. Again, that's order.allelica.com. Allelica, empowering the next generation of clinical genomics. Did you know that genetic testing can pinpoint the cause of inherited retinal disease in about 60 to 70% of cases? Identifying the disease-causing gene mutation can help individuals qualify for a clinical trial for an emerging therapy, many of which now are gene or mutation-specific. The MyRetina Tracker program offers no-cost, comprehensive genetic testing and counseling to eligible patients in the U.S. for inherited retinal degeneration. The MyRetina Tracker is designed for people with a clinical suspicion or a diagnosis of a retinal dystrophy. Blueprint's MyRetina Tracker program panel includes 351 genes and assessment of non-coding variants. The panel has excellent coverage of the RPGR-ORF15 region, which is critical in retinitis pigmentosa diagnoses. And here's a bonus, the panel includes the maternally inherited mitochondrial genome. Stay tuned for our interview with Blueprint Genetics about the MyRetina Tracker program and the patient registry being established on a future episode of DNA Today. In the meantime, you can learn more at blueprintgenetics.com. Again, that's blueprintgenetics.com. Did you know 90% of people carry at least one drug-reactive gene variant? Yes, it's actually that common. That's why Picture Genetics PGX test is so helpful. The at-home test kit helps you personalize your medication planning based on your specific genetics. The test is powered by Fulgent Genetics, a clinical laboratory that uses pharmacogenetics to analyze your genes to help you identify what meds work best for you. No more blind guesses. PGX testing gives you genetic insight on your body's ability to process different drugs. Tests come with a clinical report and genetic counseling, so you're fully supported and educated about how the results could impact you. Use the power of genetics to find the right medications with Picture Genetics PGX test. Visit picturegenetics.com and use code DNA today for 25% off and free shipping. Again, that's picturegenetics.com with code DNA today for your Picture PGX test kit. Get actual genetic insights today to benefit your health of tomorrow. Oh, and check out Picture Genetics' interview about pharmacogenomics with their Director of Genetic Counseling, Gregory Kellogg, on episode 174 of DNA Today. Another area that I kind of wanted to dive into a little bit, we've been talking about whole genome sequencing like in the clinical setting. We've been talking about it in terms of newborn screening. But what about in terms of hereditary cancer? So for hereditary cancer syndromes, those, as I've seen, when I was a student in cancer doing genetic counseling, usually that's ordered with a panel of genes that are known to play a role in the development of cancer. Now, why could it be advantageous for someone with a potential hereditary cancer syndrome to pursue whole genome sequencing? Like, how could this offer more insight than just the cancer gene panels?
So I think we have to remember that cancer gene panels, and any gene panel for that matter, is very evidence-based, right? You're looking at a bunch of genes where the evidence is pretty well established. We all know that, especially counselors who have worked in cancer genetics clinics, there are many patients who come in who have got a family history of cancer, and you really cannot pinpoint a gene on them. And in many times, we can't even find the gene. I think that's where whole genome sequencing can play a big role. That the step up from a panel should be straight to a whole genome by skipping the whole exome in the middle, because there's no point in doing the exome at that point. And this goes back to the very first question you asked me, whether you do an exome or a panel. If you do a panel in the clinic and that is negative, straight go to the genome.
Yeah, so you always have to reflex to the next testing, because if you didn't get an answer, you got to keep looking.
Correct.
Yeah, that's interesting, because I would think if we start doing whole genome sequencing as a reflex in these hereditary cancer families, that we're going to start identifying more genes to then be adding to the cancer panels as well.
Exactly.
So it kind of works backwards almost too, that it helps people in the future of, these cancer panels have really grown. It used to be years ago, for hereditary breast and ovarian cancer, we'd only screen for BRCA1 and 2. Now it's so much more than that. I'm always, telling patients, if someone in your family had testing years ago and you're saying it was negative, maybe it was just those 2 genes. Maybe that should be revisited.
And the way one of my mantras is that the genome informs the panels.
Yes.
Right? Also, the genome informs the multi-omic strategies, because one of the things that is going to come very quickly into clinical labs is functional analysis and multi-omic strategies to figure out exactly what is going on with some of the variants which we just don't know what to do with them. Remember, our diagnostic yield is still sitting somewhere as an average of exomes genome around 30 to 40%. And that is the next leap we need to take into RNA sequencing, proteome sequencing, and functional genomics.
So that, it interests me, the RNA sequencing. How does that help us understand these variants? If we have a variant of unknown significance, we say, we don't know if this is causing the gene not to work or it's just fine. It's just doing its thing there. How does RNA sequencing help us understand that difference and understand that variant?
So basically, when you are doing RNA sequencing, you're looking at the direct impact of that variation on the gene transcript and then thereby the protein itself. So there are just two quick ways to explain it. One is a variant of unknown significance. It could have a variety of effects. One is a splice site defect, that it creates a new splice site by which the gene is the transcript is truncating earlier than its usual length. There are some of the defects which is still difficult to detect, such as if the variant is causing a kink in the protein, which we still cannot see doing RNA sequencing. But RNA sequencing is probably the first step. So my own research is in neuromuscular disorders, and specifically dysferlinopathy, where we have found deep intronic changes. We have one in compound heterozygotes, where one pathogenic and one variant of uncertain significance, we have been able to show that it causes a splice site defect. So there are a variety of ways to apply it in the clinic itself.
And so is that case by case? Like, can you do RNA sequencing for a variant for a particular person? Or is it more on that research side of, now we know this variant, so now we can update clinical guidelines in terms of like what the labs are calling a variant, or, pathogenic variant versus benign variant?
So right now it is case by case. You do your usual panel exome or genome. If the variant really fits the clinical phenotype, you can look at RNA sequencing. One of the drawbacks of RNA sequencing, which we have to all remember, is that the gene expression matters. So for example, Duchenne does not have a transcript in blood. You have to do the RNA sequencing on a muscle biopsy. So you have to have a valid reason to go and do such an invasive technique, right? So,
most people don't want to sign up for a muscle biopsy. So.
Exactly. And now, you have to really say that this is really necessary to prove the clinical diagnosis. So it depends. Many people have said that, RNA-seq can eventually become the first year. But it really depends on the gene expression, which tissue is the gene expressed in. You really can't do it for intellectual disability.
Because you can't get a brain biopsy, so
Exactly.
That's not going to work. Right. So if it's available in the blood, it's easy, relatively.
Yes, yes.
So another way that we can look at figuring out variants is I think about in the pediatric setting of doing trio testing, where we test the child with the condition, or where we're trying to figure out what's going on with that diagnostic odyssey. And we say, well, let's look at both of their parents' biology and say, okay, if we find that parents have these variants and the parents don't have those symptoms, then we can say, well, it's probably not what's causing it. That helps us have information. Are there cases where we're also looking if that child has a full biological sibling? Do we ever do more than trio, where we're doing 4 genomes to look at if their sibling either is asymptomatic or if they're symptomatic?
Yes. I will tell you, nowadays we get a lot of quads, and more than that in our lab, sometimes entire families. Analysis is not trivial, but the one good thing about doing quads and more than that is, and most of these cases are cases where they have done a panel on a proband and identified several variants of uncertain significance. And I give the credit to the genetic counselor and the physician that they, from there, that single proband panel, they went straight to a genome and did a quad or a more. Because
they followed your advice. All right. Going from panel to genome.
And that's where you can rule out most of the variants of uncertain significance. So it definitely is a step up and helps. But it's also important to do the quad analysis individually also. What I mean is that the sibling against the parents and the proband with the sibling and the parents, and then take the sibling out and do the reassembly, because there's going to be a 50-50 chance here. So that analysis is involved but very important.
Yeah, it's just interesting how we can do this in so many different ways of like, how can we figure out these variants? And I think it's exciting from, a research standpoint, and just how far we've come with genetics is, we say a lot on this show that, the technology moves faster than the clinical guidelines and the insurance companies and all that can keep up with, which is exciting, but also frustrating when you're working with patients.
Yes, absolutely.
And I wish we had more time, because you're such a wealth of knowledge with this. So I'm just asking all the questions that I've had about whole genome sequencing here. I work in the prenatal setting, so I'm meeting with patients that are pregnant or looking to become pregnant in the near future. Is whole genome sequencing being done on amniocentesis, chorionic villus sampling, products of conception, so either invasive testing during pregnancy, or if someone has terminated or have a miscarriage, is this being done either during pregnancy or after?
We have just launched it just this past month, that we went live with, obviously it has to be driven by an ultrasound or some finding that drives exome or genome sequencing. So we did do a study with an investigator at Thomas Jefferson University for fetal hydrops, and that was a very high yield study. The data is just published in Genetics in Medicine.
Great. We'll put that link in the show notes for everybody.
Yeah, and it's definitely moving in that direction.
So for this, we have to see something on ultrasound that we haven't figured out through carrier screening. Is this a situation where you'd say, a standard set of tests to order for an amnio or CVS is looking at doing FISH, karyotype, microarray, AFP, which is a little bit separate, but would you say start with that, and if all that comes back negative, then reflex to whole genome? Or would you say start with that if ultrasound findings are like, just, you can't figure it out?
I would say straight go to the genome, because remember, genome is going to pick up copy number variation. So everything you're trying to detect by FISH, you're going to get identified. The only thing probably is the balanced translocations. That's
always the exception.
And I've been pushing so hard. It's really a bioinformatic exercise to get to that. But if that is not something that is your primary suspicion, I would straight go to the genome.
And during pregnancy, everything is so timely. So, it's important that, I figured you were going to say that, but you don't want to be ordering tests 3 weeks later, you get those results, and then you're like, oh, let me order a whole genome. Like, you don't really have time to play with during pregnancy. So that's a good point too.
Exactly. And if you have had an IPT done and that's ruled out as negative and there is an ultrasound finding, I think it's best to go to the genome.
Makes sense, yeah. Especially if the NIPT, non-invasive prenatal screening testing, if that has come back that it's looked at all the chromosomes. So there's some labs that offer, it looks at expanded aneuploidy analysis. So it's looking at not just the common 13, 18, 21 sex chromosomes, but looking at, any major missing or extra pieces of chromosome, which I think is helpful, because I've had that come up a couple times of a trisomy 7, a trisomy 9, something that normally wouldn't have been picked up, but we're like, oh, okay, this is making more sense.
I mean, we just picked up a UPD just yesterday in the lab.
Wow.
And that is an advantage of whole genome, right? You're just getting everything done at the same time instead of having to, and as you mentioned, time is so critical here in a pregnancy.
Yeah, certainly is. But thank you so much for coming on the show and just sharing your wealth of knowledge. I mean, you're a world-renowned geneticist, so it really is just such an honor to have you on the show. And, you know, I was looking up in the textbook that you wrote a chapter of, you're one of the editors, Modern Clinical Molecular Techniques. I was like, this looks familiar. And it was one of my textbooks in undergrad. So, and lucky listeners, you can enter a giveaway for this textbook. You can search DNA Today on Instagram and Twitter. You can also search my name on LinkedIn, Keira Dineen, and you can enter our giveaway on there to win a textbook. And I believe I was checking on Amazon. I think it's around like $200. So this is like, you know, and it's a staple textbook. I feel like anybody that has, you know, have any kind of lab background in genetics, like myself, this is just like I feel like everybody has one, you know. I'm sure I have a copy of it up here at my desk. But thank you again for coming on the show. And for people that want to learn more, they can head over to PerkinElmerGenomics.com. You know, as we've been talking about, you guys just launched the prenatal whole genome sequencing, and you guys are a global provider for just so much in genetics. So just very exciting to have you guys as a sponsor on the show, and to be providing so much education. So thank you so much.
Thank you. Thank you for having me, Keira.
For more information about today's episode, visit DNApodcast.com, where you can also stream all episodes of the show. We encourage your questions, comments, guest pitches, and ideas. Send them all into info@DNApodcast.com. Search DNA Today on Twitter, Instagram, YouTube, Facebook, so you can connect with us there. And a favor, please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allow more genetic nerds like yourself to find the show. DNA Today is hosted and produced by myself, Keira Dineen. Our social media lead is Corinne Merlino. Our video lead is Amanda Andrioli. Thanks for listening, and join us next time to discover new advances in the world of genetics.
The genes of you and me, the genes of you and me are all made of DNA. We're all made of the same chemical DNA. We're all made of